So now we're going to move away from urochordates and cephalochordates, which are two phyla within the superphylum Chordata, right? And move to the final phylum, which is Craniata. We're going to talk about Craniata for the rest of the semester, basically. And so Craniata includes all of the rest of vertebrates, excluding one group that we call hagfish, which are kind of this intermediate group or outlier, if you will, between urochordates and vertebrates. So the group of fish we're going to talk about first are called round-mouthed fish or jawless fish. And there are only a couple of species that are still alive today. But a long time ago, like 450, 500 million years ago, there were lots of these fish around, and they are often called ostracoderms. So these are the fossilized jawless fish that are found in high numbers within rock that's about 500 million years old. And so this is a great name. Ostraca means bone, derm means skin, bony skin. So they had bone, really heavy plates of bone underneath their skin. And the skin in vertebrates does give rise to bone in a number of other groups or taxa of vertebrates. And this bone is obviously typically for protection, right? It adds armor, if you will, to the outer surface of these fish. And that is separate from their endoskeleton, the skeleton that they have. And really the endoskeleton in these fish was probably cartilaginous, or is cartilaginous, at least that's what the evidence shows. And so they had a cartilaginous endoskeleton, and bone first arose in the skin. Now this bone will be modified as we move into more modern fish into what we call scales. All right, and scales are really just reduced bone, as you'll see. We'll reduce it, make it bendable, and it is still made by the skin, particularly the dermis of the skin. So ostracoderm is boned dermis, if you will, or bony dermis. And they are really, really diverse as far as what they look like throughout their kind of evolutionary history. And they go extinct because of the invention of the jaw. When fish start developing jaws, they basically outcompeted or ate all of these cyclostomes. But there are a couple groups that made it to present day. They're called hagfish and lampreys, and we're going to talk about them a little bit. They're very unique and odd of fish. So this shows you the vertebrate story, which is kind of where we're moving. Story spans about half a billion years, about 500 million years, right? Some of the most complex and largest animals ever evolved from this group, as you guys know. And this kind of shows you an abundance phylogeny of the vertebrate groups. You can see starting at about 542 million years ago up to present day. And you notice we have a bunch of names up here I'm going to go over. So we have fishes, tetrapods, anamniotes, amniotes, gnathostomes, agnathans. And we're basically going to work our way up towards mammals, and we're going to do a taxonomic review of all the vertebrates. And so we're going to start way down here with the ostracoderms and what we're going to call a group called cyclostomes. So cyclo means round, stome in Latin or Greek means mouth, so they're a round mouth. Another name that I'll show you is agnathan, which means no mouth or no jaw, if you will, as far as a movable jaw. And then we're going to move into jawed fish and then move our way up into what we call tetrapods. So, now, as you know, Amphioxus is small, right? About like this, maybe a little bigger than that. And vertebrates tend to be quite large compared to that. Really, you might wonder why Amphioxus is small. And one of the major problems with Amphioxus as it gets bigger and bigger and bigger is its surface area becomes too small for the volume that it holds. And as you push cells into the inside of the body away from the surface, it gets really hard to exchange nutrients, oxygen, carbon dioxide in those things. And remember, their respiratory pump, if you will, is ciliated, right? It's a ciliated pump, right? There's a wheel organ, there's cilia that are moving water through a pharynx. And if we just assume the pharynx is a respiratory organ along with a feeding apparatus, that really limits how big you can get because there's only so much water you can pump per hour, if you will, through a ciliated structure. So the evolution basically produced some innovation that allowed the organisms to get bigger. You can imagine another problem is the notochord. It's a nice rod. We can make it kind of harder in some species, but it really limits how big you can get, right? Before it's really hard to move the body because there's not enough surface area on it to attach muscles, for example, to move a larger animal. So one of the immediate innovations is the vertebral column, which is going to develop over the top of and eventually replace most of the notochord. And at first in lampreys, it's just simply making blocks of cartilage on top of the notochord, giving you larger body support as far as a rod and also more surface area to attach muscles. We're also going to add onto this rod spikes or spines. And the first two that evolve in all of the fossils are one that points up and one that points down off of the body of these blocks of cartilage that we're going to call vertebrae. And they're called neural spine and the hemal spine. One of the first things that happens is with the development of these spines is we're going to encase the dorsal hollow nerve cord in cartilage, which makes a lot of sense, protecting the nerves, right? Because you can imagine you could damage those really easily because nervous tissue is very delicate. And so you want to kind of encase it or protect it within tissue. And the first thing that happens is this neural spine is going to have a little arch at its base that accommodates the dorsal hollow nerve cord. So you can see here, this is what a typical fish vertebrae looks like. Here is the body of the vertebra, and it's called the centrum. The centrum is typically flat and will meet up nicely with the vertebra in front of it and behind it, right? So that they stack up like a bunch of coins, if you will. And then coming off of the dorsal side of the centrum is your neural spine. And then there's going to be a hole or pore here, or it's called an arch, which is going to be hollow and allow or accommodate the dorsal hollow nerve cord. When you stack all these up really nicely, then all of the arches line up, forming a neural canal, right? That the dorsal hollow nerve cord now can run through. The same goes for the bottom arch, which is called the hem arch or hemal arch, and you have a hemal spine. This is the ventral arch, pointing ventrally or towards the belly. This will allow for the accommodation of blood vessels that eventually are called vertebral blood vessels that actually pass along the ventral surface of the vertebral column and supply blood to all of the tissues and cells of the vertebral column. Now, as you move into tetrapods, land-dwelling animals, we are going to add some more spines or surfaces. And we'll talk about those as you move into what we're going to do next after we dissect our lampreys next week, is look at some skeletons and some innovations of the skeleton. Now, as you move into fish and then start moving into tetrapods, the middle of the centrum will be where you see the notochord or remnants of the notochord. And eventually the notochord gets smaller and smaller and smaller and smaller and less effective as you move into more derived fish, until when you get into tetrapods like us, the only thing that's left over of the notochord during development are the middle of the cartilaginous discs that sit between the vertebrae that we call the nucleus pulposus when we went over the notochord earlier in lecture. So that's the first innovation for success in size increase. The second one is we got to switch over our pump. All right, we got to get a more effective, a more efficient pump other than cilia. And so it's pretty easy to solve that problem. We know that we have myomeres, right? So this is a cross section through a cephalocordate. Hopefully you can even name some of these structures, right? Like this is the notochord. What's this thing in the middle here? That's got all these slits in it. The pharynx, right? These are ovaries, right? So hopefully everybody saw those yesterday, right? But you can see, if you follow the myomeres down, notice they taper off and kind of disappear, right? When you move ventrally, correct? As you move towards the belly of the cephalocordate. In order to kind of produce a more effective pump, what's going to happen, or what does happen through probably simple mutation, is just to increase the muscle mass so that it goes all the way around the entire body, right? And so when you look at a agnathan or a lamprey, which we're going to do a dissection of, in cross section through the pharynx, this is through the branchial region where the gills are, you can see muscle here in kind of the creamy color goes all the way around. Do you see that? So it goes all the way around the body, and so you got myomeres or lateral body wall muscle that moves around the entire body. Now what that's going to allow this organism to do is to crush the larynx or to squeeze on it, right? So you can, oops, I got a little excited and hit the button, didn't I? You can imagine that when they contract these muscles that are ventrally and laterally located, they can squeeze or reduce the size of that entire chamber that we call the pharynx, which now have gills in it. So you basically squish or crush the larynx, and then when you relax those muscles, it increases in size. And so now you can start this movement, right, which is a muscular pump, which can move a lot more water because it produces basically a vacuum, suction. We use the same thing. We move air, right? And we're using a diaphragm to do that. Fish are going to use the body wall muscles and then later on some other muscles to actually crush this part of the larynx and then expand and crush. And when they crush, they force the water out pores or slits on the lateral aspects of their body. And here's an example of one of these gill pores or branchial pores right here that actually empty from the pharynx where you find all of these gill filaments. And so now we can suck water into the mouth, into the pharynx, pick up oxygen, drop carbon dioxide, and then squeeze the water out. And then when the muscle relaxes and you increase in volume, that immediately sucks new water in, right? So this pump moves a lot more fluid than a bunch of cilia, right? Because of that, that allows for more effective oxygen and carbon dioxide exchange and movement of water so that the animal can support a larger size and more cells and body tissue. Now, interestingly, the bars of this pharynx are going to go from a connective tissue, a dense kind of collagenous connective tissue, to cartilage. All right, it's going to be replaced with cartilage. And it's going to become what's called the branchial basket. So this is the basket or the derived outer scaffolding, if you will, of the gill chamber or gill region of a lamprey. This is a lamprey skeleton. Here's the mouth up here, and you notice now why it's called round mouth, right? It's got a circular mouth. And this is where all the gills are. And the bars here, the woven bars, are now cartilage. And the reason for that is because cartilage can be crushed without damage. And when you relax, it rebounds like a stretched rubber band. So it'll rebound back to its original shape every time you crush it and help draw water back into that chamber as it expands. And so it adds to the suction ability, if you will, of this muscular pump. It also will protect the gills and not be damaged by this, crushing, relaxing, crushing, and relaxing. So it's a much more reliable tissue to have cartilage there. Matter of fact, most of the skeleton, including this basket, is cartilage, right, in lampreys. The only thing that's really ossified or is kind of bony are the teeth, right, which we'll find in the mouth here. Other than that, they are scaleless and have no bony tissue associated with their bodies. So vertebral column and a muscular pump, which will replace our ciliary pump, allows vertebrates to get bigger and to move a lot more water. Also, this is going to set us up, foreshadowing, to be able to move air. Can you imagine trying to use cilia to move air? I mean, little hairs? It wouldn't work, right? We couldn't move air. And so we still use a muscular pump, right, that surrounds our lungs to actually move air. But you also know we have lots of cilia still in the respiratory tract. So the cilia stay, but their job is going to be very similar, at least the job in our respiratory tracts, to one of the jobs or one of the functions of the cilia here, to spread mucus. I mean, that's what they do, is they move mucus. That's very prevalent within our respiratory tract for protection and to humidify air. And so you still have those ciliated cells there to move that mucus around within our respiratory tracts. The other thing that you see immediately, and hopefully you saw this from Amphioxus just to the Ammocetes, is a much more development in the head, right? Real eyes, ears, right? A pharyngeal gland instead of an endostyle. We start getting larger, more derived, more complicated structures. It's called cephalization, is the name that they use in development. The development of the nervous system in the head. Because of that, development of the nervous system and a bunch of sensory structures that allow us to interact with our environment, the head becomes more complicated, right, as you move into more and more derived fish and vertebrates. And this just shows you the cranial region from fish through mammals, showing you that the head changes a lot, right? It becomes more complicated and accommodating, if you will, for things like eyes, nasal epithelia for smell and the sensation of smell, for the movement of air and warming and humidifying of air in tetrapods. There's a bunch of sensory structures in the head in fish and reptiles and amphibians that we lack, particularly those that are involved with a semi-aquatic to aquatic lifestyle. They hear through these sensory structures we'll talk about as we move through sharks and lampreys and bony fish and even aquatic amphibians. So there's the innovation and increase in the complexity of the cranium or the cranial region of vertebrates. All right, so those are kind of some of the major innovations that happen really quickly that allow vertebrates to diversify very quickly. Within the cranium, the development of the jaw is probably one of the largest wonders of evolution that happened really early in vertebrate evolution, because that movable jaw allows them to feed differently, right, than a round suction cup, if you will, that you see in lampreys, for example. And that jaw, those jawed fish that evolved really early pretty much just outcompete and eat all of the cyclostomes, as you'll see, which disappear almost immediately when the jawed vertebrates appear in the fossil record. So here's the easiest way to classify a group we're going to call a subphylum. So we got phylum Chordata, subphylum, right? We're going to call it phylum Craniata, I guess, right? We're going to call it subphylum Vertebrata. And these are some groups, some taxa, which are mostly evolutionarily sound within the vertebrates. The first one are called tetrapods. So tetrapod, the derivative, the Greek and Latin derivatives of this are four-footed, right? So four-footed organisms are, another way to think of tetrapods, they're all terrestrial, right? TT, tetrapod, terrestrial, those that live on land. They include four major clades: amphibians, reptiles, birds, and mammals. In reality, it's amphibians, reptiles, because birds are reptiles, and mammals, but I still teach them separately. So those are kind of your 4 major groups of tetrapods. They all include all of these, and you might say, well, wait a minute, snakes don't have legs, right? Whales don't have feet, they have fins, but all of the ancestors of these organisms were 4-footed, right? So if your common ancestor was 4-footed, then you're a tetrapod, even though you may be legless, a legless lizard, for example. We also call them quadrupeds, which means the same thing. Quadro means 4, peds means feet. So it's just another name for them. Anything that's not a tetrapod, we consider a fish. All right? So fishes include 2 groups, jawless fish called agnathans, and then gnathostomes, which means gnatho means jawed, mouth. All right, stome means mouth. So and we call these the jawed fish. Tetrapods are nested in gnathostomes. We are the climax of fish evolution. I know that's weird, right? But we are nested within the gnathostomes. Now, the tetrapods and the fish, you can also break them down into 2 other groups that you might see, and they're called amniotes and anamniotes. So amniotes have extraembryonic membranes around their embryos. So they have an embryo and then they surround it with tissue. There could be several of these membranes or tissue membranes. The name amniote comes from the amnion. That's where the name comes from. The amnion is the membrane directly around the developing embryo. Reptiles, birds, and mammals are amniotes. Amphibians and fish are anamniotes, which means they don't surround their embryos with membranes. They surround their embryos with jelly, with glycoproteins, basically. That's form kind of a buffer between the embryo and the environment. So it's another way we can classify them. So I use a lot of these names interchangeably. Okay, questions? Okay, so here's our phylogeny, right? So, metazoa, remember that means tissues. We got bilateralaria, our infrakingdom, right? Deuterostomia, one of our subkingdoms. We have 2 superphyla, right? It's not written up here, but remember Ambulacraria is the name, the superphylum. And we have these 2 phyla within the superphylum Ambulacraria. We got Echinodermata and Hemichordata. So remember we talked about the hemichordates. Then we come down here and we have superphylum Chordata, right? And then we have 3 phyla, Cephalochordata, Urochordata, and then Craniata, right? This is where we're moving into now. So Craniata has 2 subphyla, Vertebrata, and then this group right here called Hyperotridae, right? Hyperotridae are the hagfish, right? Which look like a lamprey, but are a little more primitive, a little more ancestral, and we'll talk about them. Then everybody else from lampreys on belongs in the subphylum Vertebrata, or Vertebrata, if you want to say Vertebrata. So the vertebrates all have backbones. And so you can see if we follow the vertebrate line, most primitive group that we can use, or we can call the ancestral group, are lampreys. And then here are all the fishes. Lots of fish, right? These are all fish. And here are your tetrapods down here. Of course, we'll open them up eventually, right, and look at them. And don't worry, you don't need to know all these darn names over here, right? We'll learn some of them, but not all of them. All these guys down here, these are all of our invertebrates, right, on the rest of this. But we're going to open this up and start talking about the fishes. So here is an ancestral fish. This is a placoderm. It was the size of a Volkswagen. It was absolutely huge. It was the major predator of the ocean at that time. It looks pretty scary. So imagine that. You could sit inside of its jaws. So it was a major predator. This is a typical bony fish. This is a living bony fish. It's called a look down. This is just an x-ray of one, showing you the backbone, right? All right, so let's talk about this phylum Craniata, right? So phylum Craniata has two subphyla within it. So subphylum Vertebrata and then subphylum Hyperotridae. So Hyperotridae are hagfish. So this name, Hyperotridae, hypero means on top of or sitting on top of. Tridae means one piece. And what it refers to is the one nostril that sits way on top of the head. That's where the name comes from, if you want to know the Greek kind of translation of it. And if you look at this is a hagfish. This is kind of how they rest. They are called hagfish. Everybody knows what hag means, right? Ugly old witch or lady. Disgusting is another term for hag. Hagfish are really gross. They produce copious amounts of mucus. I mean, so much mucus that if you irritate them in the water, they just surround themselves with feet of mucus, and it's really slimy and nasty. And I've never tasted it, but I assume it tastes bad, right? So it's like producing really nasty snot, a lot of it. They release ink. Yeah, these guys surround themselves with slippery mucus. And so that, nothing really bothers them. So that's where the name hagfish comes from, at least. They're pretty important. They're detritivores. They live on the bottom of the ocean, and they kind of eat dead stuff. So, they're kind of gross, I guess, when you think about them as far as a craniate. Lampreys, as you're going to see, the adult lamprey are pretty scary looking. They don't look like much until you look inside their mouth and see the copious amounts of large teeth. And if you know anything about them, they are called a sanguinophore. They're blood eaters, right? They're parasites of other vertebrates. And so they are vampires, if you will, real vampires. That's what they eat, is blood. And they do that by suctioning onto a fish using these two huge teeth to open up a wound. And then they kind of rasp a sore, and they release an anesthetic and an anticoagulant, and then proteolytic enzymes that keep the wound festering, and they just suck the fish dry. Pretty gross, right? I've always wanted to attach one to me to see. I don't know. I'm weird. It'd be fun. I know. I'm a strange guy. River monsters. Have you seen river monsters? River monsters he had one attached to him. Yeah. They're not related to leeches in any way, but so it's an example of convergence, right? And the anesthetic they produce is unique to lampreys, and I put the name in the PowerPoint later on. So this is kind of a reduced phylogeny for you, hopefully to help with the names. So here is our superphylum Craniata. We got rid of Urochordata and Cephalochordata, and we just have this is phylum Craniata. I keep saying superphylum for some reason today. Phylum Craniata. And this is your subphylum Vertebrata and your subphylum Hyperotridae. So hagfish, vertebrates. All right, and then the vertebrates we can break down into lampreys and then everybody else, right? Which we're going to call Gnathostomata, all right? So, which is jawed mouth. So let's talk about these two groups. They are similar in a lot of ways. And so there's a lot of overlap with them. Now, this group that they belong to, there aren't very many of them left, and we used to use this name, this phylogenetic name. We used to use this clade name. We used to call them Cyclostomata, but we don't use that term anymore which means round mouth. I like it, cyclostomes, because it's polyphyletic, which means it has too many ancestors. There's no common ancestor to all of the ostracoderms. At least that's what they believe based on the fossil record. And hagfish are not really closely related to lampreys. If you look at them, they are very different. Hagfish are a little bit more simple and primitive compared to lampreys and the rest of vertebrates. So we don't include these in the vertebrate group. All right, we kind of give them their own kind of subphylum now. And there are a number of reasons. Number one, physiologically, they are simple. Physiologically, they are not regulators of most aspects of their physiology. Now, what I mean by that is they mimic saltwater in their osmolarity. They mimic oxygen concentrations to what's around them in the water. They don't try to regulate their body tissues and what's going on in their body tissues. You might have heard the term homeostasis before. They really don't practice regulated homeostasis like all vertebrates do. So they're very invertebrate-like when it comes to that. They also have a simpler cranial region. They have a simpler setup to their cranial region, particularly their inner ear, which we'll talk about. That's different than lampreys. And their vertebral column is less derived, right? Is less complicated than lampreys and the rest of the vertebrates. So they're often, kind of thrown out as a nice outgroup because they have a lot of the characteristics of vertebrates, but also have some characteristics of invertebrates. But we include them in cyclostomes, which makes them polyphyletic, right? If they're not sharing a common ancestor with lampreys. So we're all the way over here on our subphylum Vertebrata tree. They're going to be, lamprey or hagfish are going to be our outgroup, if you will. So now what I did is I threw this figure in because I didn't know how much anatomy you have. And I actually found this really nice 2019 Nature paper. When I first did this lecture I made this lecture a long time ago, and I've been adding to it for, like 18 years, to be honest with you. And so as new stuff comes out, I add it. So there's, they've known this for a while, but hagfish have a much simpler setup to their inner ear. So I don't know if you know much about the inner ear, but in fish and tetrapods, derived fish and tetrapods, we have this setup, the same setup that the zebrafish has. And what we're looking at, and don't worry about all of the labels. I didn't even give you all of the label names because it's not really relative to our discussion. But if you look up here, you can see that there are these 1, 2, 3 canals, right? Here's one canal, here's another canal, here's a third canal. Those are called semicircular canals. If you've had any human anatomy, we have 3 semicircular canals in our inner ear. And what do? They have fluid in them, and the fluid moves around like this, or like this, or like this, right? And so when you drop your head, the fluid moves forward. When you drop your head backwards, the fluid moves this way. When you spin really fast and throw up, at least I do, you're spinning, right? You're spinning that fluid around. And there are little hairs in those ducts that bend with the fluid movement, and then that's what you sense as your head in 3-dimensional space, right? It's basically detecting where gravity is, correct? Does the same thing in fish. They know which way's down and which way's up and which way they're moving because of the fluid moving through the canals. And most of the derived fish have the same setup we do. Now, if you think back to urochordates, they have the little otolith that does that, right? The little stone that was on a tether that was inside of a vestibule or a little space next to the brain, and that's how the urochordate knew where he was. This is a much more derived and much more sensitive system. Now, hagfish are the most primitive, or what we would call the simplest of the group. They have 1 semicircular canal. Okay, so they only have 1, and that makes them, really non-vertebrate-like. Almost all vertebrates have 2 to 3, right? Most have 3. Lampreys have 2. So here's one here. Here's the second one. They do touch, but they're separated from one another. All right, so they have 2 semicircular canals. Everybody else, all the rest of vertebrates, have 3. So that's one of the things that makes the Hyperotridae kind of primitive. So they have no bone. They're all cartilage. This is a hagfish up here. They have 1 nostril on top. That's where their name comes from. 1 vestibule. The vestibule is the cavity where the semicircular canal begins, where the fluid kind of runs to, is into the vestibule. So they have one vestibule. Everybody has one vestibule. And they have one semicircular canal, which is what is really different about this group compared to all the other vertebrates. They have no appendages, no limbs, right? And so their defense mechanism is to wrap up like a knot. They knot their body on purpose, and they pull, all right? So if you were to grab onto one of these, they used to make this toy that when you grabbed it, it was a tube that would squeeze out of your hand and you couldn't hold onto it. Have you ever played with one of those before? I can't remember the name of it. I got one for my kids. They thought it was hilarious. That's what this reminds me of, trying to grab onto one of these. They would just slide out of your hands, and they knot themselves to pull away from you. And they also they suction onto things and knot their body and pull on the knot to take chunks out of dead fish. That's kind of how they eat. And so that's kind of the way that they escape, capture food, and move away from predators. So they will do this if you grab onto them. They knot up and then pull on the knot. And they're very slick and slippery. There's no larval stage. So these are an example of a typical organism that has direct development, right? Just like you. They're born into a little miniature hagfish that grows into an adult hagfish. So there's no larval stage like mammals. They both have male and female gonads. So they are hermaphroditic or monoecious. But what's different about them compared to say, a urochordate is that only one of them is functional. So really, they're either male or female, but have both gonads. Does that make sense? So that if you're a male, the testis is working, the ovary is non-functional. If you're a female, the ovary is working, the testis is not functioning. And so they're kind of interesting that way. They don't regulate their internal osmolarity, so they are not regulators. Their interstitial fluid, their tissue fluid is the same as seawater. They function a lot like an invertebrate. All hagfish are marine, right? There are no freshwater hagfish. And they're called mud grubbers. Now that's really different than lampreys. Lampreys are regulators. They can regulate their internal solute concentration. And guess what? They used to be marine, but now they're freshwater also. You can't throw a hagfish in freshwater. You throw a hagfish in freshwater, it dies. It can't handle that big change in salt concentration because they don't regulate their body tissues very well. Well, you take a lamprey and you throw it into the Great Lakes, which has happened. That was once the marine lamprey, is now the lamprey you find in the Great Lakes. So they can live in fresh and salt water. So one of their favorite common names are mud grubbers or slimy eels. That's what a lot of people call them, but they're not an eel. They're a hagfish. So they're considered para or polyphyletic, depending on who you read. So they're going to be like sister to vertebrata or the outgroup to vertebrata. So here, look at this. These, the people who study hagfish are interesting people. So this is them playing with the mucus. It is incredible. Their mucus is really water. It has a really high water potency. When you add it to water, it expands greatly, and you only need a very little bit of water. And that's why they can produce so much of it. It's basically a water sponge. The video will show you. The video is really cool. I'll go ahead and play it. Here's this guy. He's got a hagfish. He knocked it out, and he shocks it. That's an electrode, and when you shock the skin, they'll produce the mucus, right? They excrete mucus, and he only gets a really little bit of it. So that's a little beaker of salt water. So he adds it to the water, and it's just incredible. It just sucks the water up, and when he pulls it out, you're going to be like, what the heck? So that really little bit of amount into 300 milliliters of salt water produces this kind of mucus. Isn't that incredible? And I mean, you want to play with it. I mean, it kind of looks like something it would really just make people sick, right? Disgusting.